Welcome to the All of Us Research Program Friday office hours. My name is Sam Stewart. I'm a project manager here at the Data and Research Center. I'm very excited about our presentation today. I just want to note that if you do have questions throughout the presentation, feel free to come off mute and ask or put your questions in the chat. Myself and my colleagues will help Dr. Waxse get those questions to him so he can answer throughout. I'm first going to go ahead and introduce our speaker and then pass it on over to him. So we're really excited to have Dr. Bennett Waxse, who is a clinical fellow at NIAID Medicine Pediatrics Infectious Disease Fellowship. It's always a mouthful to say that one. Who's working in the Precision Medicine Health Informatics section under Dr. Josh Denny. So if you guys don't know, Dr. Josh Denny is the CEO of the All of Us Research Program, and his team and lab group is pretty big, and they're doing some amazing work on All of Us research. So we're really excited to have this presentation today. Dr. Waxse's focuses on computational methods to extract research-grade phenotypes. So this is a great presentation because we do know that a few of you all are very excited about PWASs, phi codes, phenotypes. So this is a great presentation to learn more about that. So he did recently publish this work on computable phenotypes for respiratory viral infections that enable the precise identification of episodes using all of us data. So he's currently applying these methods to investigate host-pathogen interactions through GWASs or genome-wide association studies and clinical risk factors. And so I'm going to pass it over to him, and then I will plug in the publication information while he's presenting. So go ahead.
Okay, I'll get my slides. All right. You can see, yeah, I think my presentation is up. Okay. Thank you for the great introduction. I'm Bennett. I'm an infectious diseases fellow. I am very grateful to be invited to come speak. I have been an ID fellow for 4 years. I've been working in Josh's lab for 2, and I have really thoroughly enjoyed learning how to phenotype in the electronic health record, specifically in All of Us. And so we're going to talk about respiratory viruses. A quick note that we sought resource access board approval for some numbers reported that are less than 20, but they decided that exception was not needed and that it's all in compliance with their policy. And I will start with an acknowledgement for the lab. So Dr. Josh Denny's lab is a great place to do research. I've really enjoyed being here. Underlined folks are people who have really helped with this project. But I'm also grateful to collaborators I have at the Uniform Services University, Dr. Ricotta and Bustos Carrillo. And then special thank you to NIAID, my program director, Dr. Krista Zerby, and DIR director, Dr. Steve Holland, who let me leave NIAID to do research at a different institute. Of course, we should always start by thanking also the All of Us participants, because we couldn't do this work without the more than 800,000 participants that make this possible. So I will outline this talk. We'll start with some background about respiratory viruses. And then we will dig into the phenotype that we put together. So this is, I'll show you how we identified common respiratory viral infections using All of Us, and then how we compared these results to epidemiological data from the CDC to verify that the results that we're finding in All of Us match what we expect for national data. Then at the end, I just have a few examples of applications of how we're looking into these phenotypes to tell us some interesting things about long COVID, genetic susceptibility to influenza and other viruses. So I don't have the chat open, but I'm very open to questions throughout. And so if anyone has any questions, feel free to interrupt so we can talk about this along the way. But let's get started. So first, some background. The respiratory viruses are, I'm going to say, not just a common cold. So they are the most frequent agents of disease in humans. There's near total seroconversion. So if you look at antibodies for people who has been exposed to influenza, RSV, human coronavirus, nearly everyone has been. And this causes significant morbidity and mortality at the extremes of ages, really, but also within other risk groups, people who have chronic comorbidities, people who are immunosuppressed, et cetera. And just to give you a sense of the scale of the problem, this is some information from influenza, of course, before COVID, the most studied respiratory virus. If you look at the global burden of influenza in 2021 for adults, this was 32 million episodes and nearly 6 million hospitalizations. And if you factor in for a US burden of economic burden, if you factor in hospitalization, lost productivity, lost lives, in a single year, they estimate $35 billion. And this was all before SARS-CoV-2 entered the scene and we all endured the COVID pandemic. So beyond these viruses, there are over 25 that we know are linked to pneumonia. And if you look at hospitalized patients, there's actually little difference in the mortality rates, how severe the infections are, and the cost for these patients. And so I'm going to call this an under-recognized and understudied area of medicine, these non-influenza, non-COVID viruses. And I don't need to remind you that these are agents of pandemic potential. So the ability to have EHR-based phenotyping that could allow us to describe the biology and risk of these agents quickly would be really helpful. So we're going to take advantage of something that's inherent to respiratory viruses, and that's that they are seasonal. And so this on the top shows all the viruses that we're going to talk about today. These viruses are commonly screened and tested on something called multiplex PCR testing, where you can test for multiple viruses at the same swab that goes into the nose. And on the bottom, I'm showing that this is CDC data. So the NREVSS is a surveillance system where clinical laboratories from around the United States report their results to the CDC. The CDC makes all of this available, and they use this to track annual, monthly trends in respiratory virus transmission. And this is just 1 year's data from 2018 to 2019. And on the y-axis is percent positivity, and on the x-axis is time, just the months of the year. And you can see two things. So RV is rhinovirus, and influenza are the most commonly detected virus, just based on percent positivity. The other viruses are less commonly detected. But and you can already start to see this, but if you scale these trends to their maxima and you normalize it, you can see that they actually look like they occupy a certain part of the year. And so apart from adenovirus, which is usually bimodal twice a year, doesn't have as strong seasonality. The other viruses do. So rhinovirus usually starts off our respiratory seasons in September to October. There is usually a second peak that happens in the spring, but the biggest peak happens earlier in the fall. Usually gives way to parainfluenza, and then RSV. So respiratory syncytial virus is one of the biggest viruses that I would say we know clinically because it has importance in terms of who's hospitalized, especially people who are of advanced age or are very young infants. After RSV, which occurs around December to January, human coronaviruses, which usually are the common cold, present in between RSV and influenza. Influenza season is usually January to February, and usually followed by a virus called human metapneumovirus. It's a virus that's very similar to RSV, but you can see that as the spring turns to summer, the respiratory virus ends, and overall, over the summer months, we have very little transmission of these viruses. And so our question is, as we try to identify these viruses in all of us, can we actually take advantage of the seasonality of these viruses to tell us whether our phenotyping is working or not? Because the overall question, when you don't have something like a gold standard and a clinical note to adjudicate your phenotype, is how can we use other methods to say whether our phenotype is working or not? So that was really the big motivation for this work. For the phenotype, as you know, all of us has a lot of great data available: survey responses, physical measurements, electronic health records, whole genome sequences, and wearable data I have here. The phenotype is, of course, rooted in electronic health record data, and we'll use it to intersect with some other types of data later. But now we'll get into the phenotype. So the paper was out in May of this year. The PMID is up in the top right. The notebooks that define the phenotype are in a community workspace, which has a QR code at the bottom. And so you can use these phenotypes. The community workspace is V7, but switching it to V8 is not too tough at all. And so in this figure, I'm going to show you how we combine these elements. And this isn't a new idea. There was important work done 8 years ago, showing that when you combine different types of data, your ability to predict illness is better. And so we are just using that information to create a phenotype for respiratory viruses. So On the left, you can see three things that we denote to be episode defining. It can be an ICD code that's virus specific so an influenza code, not something like influenza-like illness, but if it's a specific code to the virus, we include it. For certain infections, influenza and SARS-CoV-2, the virus that causes COVID, there are specific antivirals that are only used for influenza or COVID. And so because they're specific to the virus, we've included those medications here as some indication that someone might have that infection. And then our best measure of someone having an infection is a test result. So NAAT is nucleic acid amplification test. It is not necessarily a PCR, and so that's why I use that abbreviation. It's the most common test that we have in this data set, and it is routinely used. I referenced it earlier as the multiplex testing. We do have some tests that are antigen, which is a protein-based test, and culture, which is where you cultivate the organism in the lab. But the majority of our tests are these PCR-like tests, which are very good and specific to the virus that they're identifying. I have one more piece of data that we'll see, and it's this open tan circle, which just denotes a negative or indeterminate test. And I'll show you how these all come together. So here on this plot, I have time zero denoted at the circle. Multiple rows are synthetic people. None of this is real data. And for instance, these two people had an ICD code on their time zero. So their first piece of data was an ICD code. This person had a medication. This person had a positive test. These people had, for instance, an ICD code and a test, or this first person had a medication, an ICD code, and a test. For people who have a negative test result, you can see that it doesn't cause a time zero, and you'll see what this row means later. So for everyone else, all subsequent information that's related to this episodic disease is probably related to the first instance of the disease. So say you are hospitalized for influenza and then you follow up with your PCP later. You might get another ICD code for influenza later. And so in this phenotype, we wrap up all concordant information within 90 days. And so you can see here, this top row person, they had a test, an ICD code, and a medication for all on the same day. And then for 4 subsequent days, they got an ICD code and a medication. And that would make sense, for instance, for influenza, where we treat for 5 days, you might be hospitalized for 5 days, et cetera. And then this person, so their negative test did not count as a time zero, so it comes before, but later they got some ICD codes in, and sure enough, they did have a positive test later. And so what I've shown here in the gray boxes is how we define episodes. So we include a negative 5-day look back window just to wrap up negative tests in case someone wants to study false negative test results. But generally, if you have a negative test, you would not be included in the phenotype. And the reason we think this is powerful is that you can do the same phenotyping approach and you can apply it to all the respiratory viruses together. So this way we can study all of these viruses in the same cohort using the same method, and that will allow us to infer clinical risk factors that are specific to these viruses using a concordant data set. So this is the overall phenotype, which we're going to identify participants with. So here is a map showing all of us participants who have relevant data. So relevant data to this talk means an ICD code, a medication, or a lab test. And this shows, what I'm showing here is comparing it to where the CDC data that I mentioned earlier are reported from. So all these black dots are CDC labs that report to the NREVSS. And you can see overall that both of these give national coverage. By no means is it complete coverage of the United States. You can see regional variation throughout different states, but both samples are sampling the whole country. And so we wanted to see what the lab results in all of us in the CDC were like. So here I'm going to show 4 plots of the same thing, just to orient you to the plots. We have all of us data, which will always be in gray. We have CDC data, which will always be in blue. The lines correspond to the left y-axis, which is % positive. So the number of test results that are positive out of all the test results sent. And then in the shaded regions below, this is total tests sent. And you can see that this corresponds to the right y-axis, And the numbers, the absolute numbers, are much different. Of course, all of us is 200 times smaller than the national CDC repository here. And so those are scaled to show similar trends. But overall, you can see the number of tests sent fluctuate year in, year out. And so let's focus first on rhinovirus on the top left. First, we'll look at the shaded bars, and you can see as respiratory season shows up, the number of tests providers send goes up. And during the summer, fewer people are sending tests. And if you look at the test trend for all of us versus CDC, they match really nicely. If you see data end, it's because I only was able to get CDC data up to that point. So now let's look at the percent positivity. That's the lines. The gray line is all of us, and the blue line is CDC. And you can see, just like I mentioned earlier, rhinovirus is showing 2 peaks per year, a smaller peak in the spring, a bigger peak in the fall, and every year it's following the same trend. And generally, the all of us data, although it's more granular, there's more variability, matches the CDC data. And we have the mean adjusted error here on the top left. Rhinovirus has the highest error, but generally for the other viruses, they're quite low. So for RSV, for influenza, for SARS-CoV-2, you can see that the test results from all of us, gray, match CDC quite nicely. And here we can also use the COVID pandemic to ensure that our tests are correct. So after February of 2020, look at the next year. So test burden went up, and both in the CDC and all of us data, providers thought RSV was going to be present, but no percent positivity rose until later in the year. And this is exactly as expected. It's already been reported that during COVID, there was not transmission of RSV and influenza, and our data matched that. So CDC percent positivity did not rise, and all of us percent positivity did not rise. Interestingly, rhinovirus was still transmitted. By the time we had this result, it was already published. It was no surprise. But of all the respiratory viruses, actually rhinovirus transmission persisted, even though influenza and RSV stopped in the setting of all of these mitigations we were doing during COVID. So this gave us the sense that the temporal changes in lab results in all of us matches CDC. So we feel confident about the lab results in all of us. Another way to look at this is to aggregate all the test results for a certain time period. So in 3 cases, we have in blue a published study from the CDC where they showed over the time period labeled in ABC, so for parainfluenza, for instance, 2011 to 2019, the breakdown of subtypes of parainfluenza. So there's 1, 2, 3, and 4 for parainfluenza. And when you compare all of us, which will be a color that's not blue, to the CDC data, the overall breakdown of parainfluenza 3 versus parainfluenza 4, for instance, matches nicely. If we look at human coronavirus, there is a flip in terms of what is the second and third most common. In all of us, we had HKU1 as the second most common versus NL63. But of course, these cohorts are different. And overall, the groups match up quite nicely. So influenza A versus B, the breakdown is very similar. And we didn't have any subtype information from influenza to compare to. But this showed us that temporally, the changes matched, and in aggregate, the changes matched. And so we felt good about the lab results in all of us. And now we're going to use the lab results in all of us as our reference standard. And we're going to bring in other types of information and see what they look like. So here, now in blue, so we no longer have CDC data in these plots. This is just all of us data. In blue, test results that rise and fall. And for this virus, we're looking right now at respiratory syncytial virus. So we're going to be comparing the other phenotypes to blue. And so in this case, you can see a light orange is 1 ICD code, gray is 2 ICD codes, dark orange is 3 ICD codes, and then a thick gray line is all combined together. And all this is to show is that we know that test results rise and fall like CDC data. Now, how do ICD codes work? Because we might suspect that people don't use ICD codes accurate, or maybe they're present during the summer, or maybe they continue during the COVID pandemic. So here you can see overall, whether you are an episode defined by 1 ICD code alone, 2 ICD codes alone, or more than 3, you rise and fall with test results. And overall, the overall detections match the respiratory virus season that we expect for RSV. We can zoom in again to COVID, and find that almost no detection was found when we knew that there wasn't transmission of RSV. And then later, when there was an out of season rise in RSV results, we did detect it. And interestingly, actually, so if you zoom in at this blue line, there's actually an increase in all the tests positive cases that arise before ICD only cases, which I think makes sense. When you have a virus transmitting outside of expected time, providers aren't going to be thinking about it. But only when you get back into respiratory season or people have been seeing that the tests are positive, they might be coding for RSV alone. And so this is our first look at RSV to see that the ICD components predict test results well. Now we'll look at an example where it didn't work. So as you might have guessed, human coronavirus codes lose their specificity during COVID. So if we look first at 2018, 2019, we can see that lab tests rise and fall during the winter seasons. One ICD code episodes are present, although they're few in number. But then when we go to 2020, we see again the same trend that we saw in all prior years. But as soon as the COVID pandemic starts, you can see a huge takeoff of 1, 2, and 3 ICD code episodes without any rise in tests. And so this shouldn't be a surprise. Before we had an ICD code for COVID, it made sense that providers were looking through the ICD dictionary, found something like coronavirus, and applied that to their patients. And so this is to show that it's really helpful when you have a temporally changing disease to look at the data over time. Because if we would have just used this phenotype as is, we would have captured a huge number of cases of COVID that were not human coronavirus cases. And so let's take a look at influenza. So because we have antivirals specific to influenza, we have two new colored lines. The green line is where you have a medication only. The pink line is where you have a medication and an ICD code. And we're seeing how these change over time. And overall, look at 2018, all of these lines rise and fall together, and they make up an influenza virus season that makes sense and is what we'd expect. If we zoom into this section, though, we can start to see a few granular differences. So first, I'll draw your attention to the end of 2020, when we knew that influenza was not transmitted. We don't see any blue lines, so we don't see positive test results, but we do see a peak arising where providers were giving patients ICD codes for influenza because they thought it was influenza season. They present with symptoms that look like influenza. They give them an ICD code. But interestingly, 2 and 3 ICD code cases are not found at all. And so it also brings up an idea that ICD codes for these different respiratory viruses behave differently. So for something common like influenza, providers might code for something like influenza without having evidence that it is influenza. Whereas something like RSV, you may only code if you know for sure they have RSV. So looking temporally, we already start to identify some areas where this might be problematic. How are some other ways that we can look at these results? So instead of looking over time, we can also look at how the phenotype behaves in aggregate. So I have two plots shown here. On the y-axis, we have positive predictive value and sensitivity. And just to remind you, I have the confusion matrix on the bottom. Here we're using lab test as our reference standard. Because lab tests are rarely falsely positive, we are using them as the reference standard. And we're asking, how does the phenotype described by things like ICD codes and medications predict the lab results? And so we're going to use lab tests as a reference standard, and we're going to say our positive predictive value, our ability for our phenotype to identify the lab result over all its positives versus its sensitivity, its ability, how many of the positives it detects. How do that change as we change the phenotype? So on the top left, we have positive predictive value. I have this rather than sensitivity because the sensitivity of all of these is about 99%. Of course, positive predictive value is subject to prevalence of disease, where sensitivity is not. So this is a much more informative measure, but what you're going to see, we'll start on the left. So 0. If you have an ICD count of 0, it means that your phenotype is positive for something else. And so that will only work for influenza and COVID when we have medications to indicate an episode. And so this is where you have influenza, you have one medication for influenza, and you have no ICD codes. That positive predictive value is poor, 60%. So 50% is a coin flip. this is marginally better. And for a disease where people can get medications in a preventative way, this I think makes sense. COVID, if you have only a medication, you have no ICD codes, is actually decently predictive. So nearly 80% predictive. If you are getting a COVID medication, that you will probably have a positive COVID lab test. And then we'll go from 0 to 1. So now we're adding an ICD code. And I think something interesting happens. So let's focus on influenza and COVID, the orange and blue. The overall positive predictive value of one ICD code alone is about 70 something percent, 75%. But if you add a medication to that, the positive predictive value goes to up around 90%. And so we can already start to see combining these types of data together can give us different cohorts with different predictive power. So that was a little look at COVID and influenza. Overall, the trends, I put here 1, 2, 3, 4 ICD codes because the idea usually is that when you have 2 or more ICD codes, the chance of having that disease is higher. But we know that these are very specific codes. And if you look at all the other respiratory viruses, they're near the top. They have a positive predictive value generally above 90%, and it doesn't look to change if you go from 1 to 2 code. So these are infrequently used codes. They don't really change as you increase. But if we look at this dark green line, that's human coronavirus. I already showed you that we know that this code was disrupted during the COVID pandemic. And if you choose in the phenotype to not allow ICD codes after COVID to be present, you can see that this trend returns to the trend as you expect for a non-influenza, non-COVID virus. So just to show you, this is without filtering. If you filter to remove after COVID, this human coronavirus positive predictive value is very high. And so we can use this to identify how to shape the phenotype. This is influenza and COVID. And so let's go back to where we were looking at 0 ICD codes, where you're indicated by a medicine only, or say here, maybe 1 ICD code only. If we remove those 2 definitions from the phenotype, so if you have 1 ICD code only, you're removed, or you have a medication only, you're removed, you can see what this does to the overall trends. So let's focus, if you can, to the end of 2020, before 2021. Remember, we talked about this peak of 1 ICD code only. You can remove it, and your phenotype is much tighter to what you'd expect. And you can use, looking at these trends to tell you how to choose your phenotype for your research study of interest. I'm going to skip a deep dive into the sensitivity, but I think this is a cool reminder that if you go from 1 to 2 to 3 ICD codes, so you make your threshold more stringent, you have at least 2 ICD codes, it just shows how you lose sensitivity. So if you go from 1 to 2, for instance, in influenza, your sensitivity goes from somewhere like 65% to 40%. And so overall, we present this in the paper just so people can decide how to construct their research questions. We have a huge table at the end for influenza and SARS-CoV-2, where we made all kinds of different combinations of ICD codes and medications and reported both sensitivity, PPV, and add a coefficient called the Matthews correlation coefficient, otherwise known as phi, which is a measure taking into account all 4 components of the confusion matrix. You can ultimately decide, do you want the most sensitive phenotype, or do you want the highest positive predictive value, or do you want the best phi? At the end of the day, like I've been saying, it depends on the research question. And all of that's available in the paper, as is this figure. This just shows another breakdown in aggregate of the phenotype by virus. So each row is a virus. Each column is a different way you phenotype, and they are exclusive columns. The middle column is total number of episodes. So you can already see for COVID, we're in all of us detecting 63,000 episodes of COVID. For influenza, 28,000 episodes of influenza. The heat map shows 100% based on the row. And so this is a figure to show by virus what types of phenotyping gets someone indicated for this infection. So for instance, rhinovirus and human parainfluenza, the intensity of the 1 lab only column shows that a majority of these patients have just a lab test. No one's really using the ICD codes. Most are identified via lab test. So of the 2,800 rhinovirus cases, 2,200 are by lab test. Compare that to flu, where nearly 10,000, about a third of all cases, are a single ICD code only. So if you remove one ICD code only, it does affect your power, but as we talked about, it changes your accuracy as well. So we'll take 1 second to look at looking at severity of infection across these viruses and according to the phenotype, and then I'll show you a few applications. So this shows total counts of episodes of each infection with severity of infection proportional to the intensity of the color. So the darkest color is an inpatient stay, lighter is ER, and the lightest is either unknown or outpatient. And I have it sectioned into whether or not you have a positive test in your phenotype. So the top row has ICD codes and a positive test. The bottom row has no positive test. Overall, I'm including this just to show us, look at the y-axis limits. They're different for COVID and flu. COVID, even though it's only been around well, in this data set, 3 years, dwarfs influenza and all of these other viruses that we detect by a significant margin. And overall, I think for something interesting, I'm going to advance this slide 1. So instead of comparing this according to absolute number of counts, we can look at it by percentage. And so if we are now comparing like intensity of visit by respiratory virus and by phenotype, as we go, like look at the far left. This is rhinovirus. We're going to go from 0, 1, 2, 3, 4 ICD codes. As you go from 0 to 4 ICD codes, the fraction of your visits that are deeply colored, i.e. inpatient, go up. And that trend applies to every virus. And so this tells you a little bit about your phenotype. If you want something that's more of a hospitalized cohort, you might require them to have more ICD codes that are positive. But it also tells us something interesting about what we're capturing. So let's compare COVID and influenza. These have a good mix, actually, of inpatient, ER, and outpatient stays. When we compare that to all of the less common viruses, which look predominantly inpatient. And so we have to be careful. Of course, right here, we're not saying that these viruses are more severe. They cause more severe disease. But actually, we're detecting more severe disease. And so this is a setup to show that this phenotype would not be used as is to say anything about the severity of these infections. What it will say is our phenotype is really good at detecting inpatient stays for these other viruses. And so that's going to frame how we do our studies in the future. And if you want to know something about outpatient rhinovirus or RSV, this isn't the phenotype to do it. So that's the phenotype. That's comparing it to CDC data. Now let's take a few minutes to look at some fun applications. So we've made this research grade phenotype. Now how can we use it to extract clinically meaningful information? So these first few are just plots that are fun. They're no self-contained research study. Just like we looked at hospitalization for an infection, on the top right, I'm showing you the time 0 of their infection. The 5 days before is that look back window. It can go up to 90. What we can also look for is, since we can just test every virus at the same time, we can say who had an episode that started within 14 days of another episode. And we can start to make research questions that have to do with co-infection. So which viruses typically are found to be co-infected with other viruses? So here, the intensity of the heat map just shows frequency. The highest co-infection was influenza and COVID, which dwarfs all the others, but generally influenza and rhinovirus showed the highest frequency of co-infections, which makes sense because they're the most common viruses. So the phenotype allows you to do co-infection studies. We looked at medication use for all of the antivirals for COVID, and we looked at them over time. So this measures remdesivir in blue, which was the first drug we used to treat COVID. And then the emergency use authorization for molnupiravir and Paxlovid, which is nirmatrelvir with ritonavir. After the EUA, you can see the switch of using an IV-only drug, remdesivir, to an oral drug like Paxlovid as Omicron took over in our data. So all of us gives great access to medication use, and you can see here trends in different antiviral use over time. This is a short shout out to a project I have with Evelyn Fulda, a graduate student in our lab, who is focusing on developing expertise in wearable data in all of us. And she has some cool work of her own looking at chronic disease and other diseases affected associated with step count. But we wanted to ask, what about these episodes? Because we have information of when people are infected, we have their step count data, and as I already suggested, we have 63,000 cases of COVID. So here we just have of all the people who had a COVID infection and or were tested for COVID, this is their wearable data. So steps on the y-axis, time on the x-axis. 0 is when they had their infection. You can see there's a big drop in number of steps. They recover their steps. And for people who are COVID negative, of course, they're still sick because they're getting a test. And so it makes sense that their steps also drop. And none of this is too interesting, but we wanted to say, what about the step data and its ability to predict someone who might have long COVID? So here we looked at, instead of COVID versus negative, we looked at everyone who had COVID, those who had long COVID after, and those who did not. And again, at 0, we have when they had a COVID infection. Here on the y-axis, the y-axis, we have steps, and that's the 7-day rolling average of daily steps. We have control patients, people who did not have long COVID in blue, and we had in magenta the number of people who did have long COVID. And interestingly, we were shocked to see, I think we expected after you would have long COVID to have lower steps because long COVID is defined by malaise, post-exertional malaise, you have brain fog, and other symptoms that make sense that they would impair your step count. And indeed, there's an MPJ Digital Medicine article that suggests the same thing. We were shocked to see that the steps before an infection were already lower for people who had long COVID. And when we saw that at first, my first thought was, okay, the people who get long COVID, we know increased rates if you have COPD, chronic lung disease, diabetes, or anxiety or depression. And so are those just explaining our difference between steps and people with long COVID and not? And so we did a model where we predicted your odds of having long COVID versus not per 1,000 step increase. So as your steps go up, your risk of long COVID goes down according to this model. You can see everything we adjusted for on the top. These are all things known to be associated with long COVID. And even when you adjust for all of these different risk factors for long COVID, the risk is still down. So 90% odds ratio of having long COVID, or I guess, yeah, 0.9 compared to 1 for every 1,000 step increase. And when we add in certain comorbidities, COPD, depression, anxiety, all of them, we're not able to erase this difference. And so this is something we are close to writing up, but a fun example of what you can do with the phenotype and wearable data. The last application I'll talk about is a brief one where we look at the genetic susceptibility for influenza. So despite influenza being important and a lot of data being known about influenza, the genetic risk of influenza is mostly understudied, I would say. There's a lot of basic science work and a huge mountain of research that we know. But in terms of assessing populations that have influenza and not, there's not a lot done to look for genetic risk. There are some small studies where they have looked at pandemic flu and found some risk variants in certain ancestries, but when they look at other predicted ancestries, they haven't been present. This study that I present here, which was in Nature Genetics, was the first gigantic study to look for genetic risk factors for influenza. And I highlight the section that I really like. So genetic risk factors are well characterized for COVID, but not for influenza, with the largest published genome-wide association studies, including more than 2 million for COVID, or about 1,000 for influenza. So that difference is true. And so what they did is they looked at ancestry DNA customers who said that they had a positive flu test result in 2019 to 2021. They aggregated 18,000 cases and compared that to 276,000 controls, people who said they didn't have influenza during these 2 years. And what they did was compare all of these risk loci from discovered in COVID to influenza. So here in red is going to be the COVID results. So the odds ratio for the genetic variant is on the y-axis. Every spot on the x-axis is a SNP associated with disease in COVID. They were discovered for either hospitalization or infection. And you can see that the risk loci show an increased or a decreased odds of risk for COVID at every step. When you compare that to their cohort, so they did it for influenza, not one measure showed a departure from an odds ratio of 1. All of these overlapped. And so this was their conclusion to this was that using this phenotype for influenza, the risk loci for COVID are not the same for influenza. They did go on to find 2 new loci. These 2 SNPs were found to have a decreased odds of influenza according to their phenotype, and they replicated that throughout a few other biobanks. But we wanted to ask, if we know more about the severity of infection, and we're not just asking, do you have flu or do you not, how does that change the results? I'm going to skip going through detail through the individual SNPs. They're in this Nature Genetics paper. They have putative roles in sialic acid linkage for which is important for influenza infection. And they talk about the enrichment in EQTLs, et cetera. But in the interest of time, we can skip over that. For infections, it's important that you know whether someone was exposed. Because if they have a risk variant, but they haven't been exposed to what will give them an infection, you can't know whether that risk variant will increase their risk for the infection. And so for infections, the basis of the phenotype is we want to know who was infected and who was not. And so we are, because the question would be, if you are asking in a survey who had positive influenza and not, it doesn't necessarily mean severity. It might say who's wearing masks, who's getting tested for influenza, who is seeking health care in that setting. So here, we think this phenotype will be better defined for really precisely saying you had influenza, you were inpatient, you were not, and can we find some genetic risk? So just to show you how we've done this on a first pass, we took 13,000 influenza cases, and this was the phenotype where we didn't include one ICD code and we didn't include a medication alone. That gave us a positive predictive value of 80%. We took these 13,000 people. If they didn't have a visit type, or we couldn't figure out if they were inpatient or not, we excluded them. If they were co-infected or if they were immunosuppressed, we removed them because maybe the co-infection or the immunosuppression is why they had a severe infection. Some people were reinfected. And so we end up with this cohort on the far right, which shows that we can, using this phenotype, get about 7,600 patients who were immunocompetent, not infected with any other viruses, and had an influenza infection. And here on the bottom, you can see the percentage breakdown. A fifth were inpatient, 25% were in the ER, and the rest were outpatient. And so our design for this study, I'm just going to show you our results on the top compared to the results from the paper. We looked at all of the COVID risk variants. We did it in an ancestry-specific manner where we looked at African, admixed American, or European predicted ancestry. We ran GWAS to look at these, we ran GWAS and then we looked at these loci. And here you can already see, generally our results agreed that for a severity of influenza, we did not replicate a lot of the risk loci for COVID. We did see 2 cases here in admixed American and here in European ancestry where we see replication of or at least departure from one of a COVID risk locus. And then on the far right, we have the 2 SNPs, discovery SNPs from that paper. So when we look at ancestry specific, we see actually an opposite result. So our European ancestry group had an increased odds ratio of severe influenza compared to the decreased risk that they published. And so we're already starting to see if you phenotype differently, when you ask a question about severe influenza versus an influenza positivity during a pandemic, you start to get different results. And we tried to replicate a closer phenotype to theirs where we just looked at flu positive versus negative. You can see that it's a bigger cohort, the confidence intervals are much lower. We actually do replicate their findings in that those ancestry-specific loci no longer depart from an odds ratio of 1, and our effect size for the variants that they identified flipped, and we could see that our risk variant was actually decreased risk now. So this is preliminary work. I think it suggests that knowing exactly how you phenotype is important when you do genetic-wide studies. And even though we have a drastically smaller cohort here, we are able to replicate some findings given our precise phenotyping. So the future directions of the work are looking at that long COVID study, writing it up, and really fleshing out our modeling. I really, as an infectious disease doctor, love the idea of wondering if there are post-infectious complications to other respiratory viruses. So that's an extension of that work that I'm really excited to do. Like, if you have influenza, are there people who have long COVID-like symptoms after? We are excited to do that with wearable data using all of us. The other two studies that we have planned are we're going to look at clinical and genetic susceptibility to other viruses. So we're going to do different combinations of influenza, rhinovirus, RSV, and others. And the power might ultimately not be big enough, but we might be able to do combinations, for instance, non-influenza viruses, to see if they have risk variants that are similar or different. So with that, I'll end and again say thank you to everyone in our lab. And if there are any questions.